Okay, I'm going to get started back up again. So the first thing we're going to go through is what happens just in terms of the electrons flow and that electron transport chain that we just talked about. This is going to start with either NADH or FADH2. And the path of electron flow is slightly different between the two. So what we're going to see is that the electrons that NADH has is going to pass its electrons initially to the NADH dehydrogenase complex. And we said that this complex pumps hydrogen ions into the intermembrane space. The electrons from NADH dehydrogenase complex are then passed to ubiquinone. And then from ubiquinone, the electrons are passed to the cytochrome C1 complex, which we said also pumps hydrogen ions into the intermembrane space. And then the electrons from the cytochrome BC1 complex are passed to cytochrome C. And then from cytochrome C, the electrons are passed to cytochrome oxidase. And cytochrome oxidase, we said, also pumps hydrogen ions into the intermembrane space. And then the final electron acceptor is oxygen. So the electrons coming from NADH in this pathway ultimately end up, and what happens is that when you add them to oxygen, you're going to create water. So the water that you produce as a byproduct of aerobic respiration is going to be produced in this last step when oxygen acts as the final electron acceptor. Okay, so that's the path of electrons in terms of going from NADH to ultimately oxygen through this electron transport chain. Okay. Now, from FADH2, what happens is FADH2 does not pass its electrons to NADH dehydrogenase complex. It first passes them to succinate reductase. So in this pathway, again, the electrons from FADH2 first get passed to succinate reductase. And then what happens is succinate reductase will then pass them to ubiquinone. So I just drew an arrow up to ubiquinone from the previous pathway. And then the pathway is the same. Okay, so from FADH2 they get passed to succinate reductase. Then from succinate reductase they get passed to ubiquinone. Then to the cytochrome BC1 complex, which pumps hydrogen ions into the intermembrane space. Then the electrons are passed to cytochrome C, then to cytochrome oxidase, which pumps more hydrogen ions in intermembrane space. And again, the final electron acceptor is oxygen. So the oxygen is the final electron acceptor regardless of whether you're getting electrons from NADH or FADH2. Okay, so that's the flow of electrons that are occurring in terms of electrons going from NADH or FADH2 to oxygen. And then you can see that you're getting a hydrogen ion concentration gradient created during this flow. And again, that's important because that's the energy source for ATP synthase in this process. So ATP synthase is not a part of the electron transfer. So electrons never get past ATP synthase. But what happens is ATP synthase uses the hydrogen ion concentration gradient that you make in this pathway to produce ATP. And specifically, the hydrogen ion concentration gradient is the energy source. Okay, so what's going to happen in the final step then is that the hydrogen ions that you pumped in the intermembrane space are going to flow back into the matrix from the intermembrane space. So are going to flow from the intermembrane space back into the matrix through ATP synthase. And again, that's going to be powering the synthase to synthesize ATP. Okay, do you have any, so I'll go through it again using the slide which has a picture with all the components written on it, but do you guys have any questions as of right now? And remember, the NADH molecules and FADH2 molecules that are being used in this final step, again, are coming from the previous stages. So we produced NADH during glycolysis. We also produced NADH during pyruvate oxidation, and then again in the Krebs cycle. And then the Krebs cycle also produces FADH2, and all those molecules are going to give up their electrons in this stage to that electron transport chain. Okay, so let's look at an actual drawing of this process. I'm going to fast forward to this slide here. So in this drawing here, this is the inner membrane of the mitochondria. So this membrane here is the inner membrane of the mitochondria. This is going to be the intermembrane space, and then this is the mitochondrial matrix. You can see that here are the different components of the electron transport chain. They're either embedded in the inner membrane or associated with the inner membrane. And then also ATP synthase sits in the inner membrane. But just remember that ATP synthase is not a part of the electron flow, so electrons never get past ATP synthase. Also, when you're looking at this picture, the red line is showing you the path of electrons, which we already wrote out on the board. And then in terms of the blue straight lines are the direction of hydrogen ion pumping. And then the dotted lines are showing you that those hydrogen ions that are pumped in the intermembrane space can flow back through ATP synthase and power that synthase to produce ATP. Okay, so again, we'll start with NADH. It gives its electrons to the NADH dehydrogenase complex, which pumps hydrogen ions in the intermembrane space. The electrons are then passed to ubiquinone, then to the cytochrome BC1 complex, which pumps more hydrogen ions. From the cytochrome BC1 complex, the electrons are passed to cytochrome C, then to cytochrome oxidase, which pumps more hydrogen ions. And then cytochrome oxidase gives the electrons to oxygen, and in that step you create water. Okay? And then from FADH2, rather than giving its electrons to the NADH dehydrogenase complex, FADH2 gives its electrons to succinate reductase, and then succinate reductase gives them to ubiquinone, and then the pathway is the same. From ubiquinone, they get past the cytochrome BC1 complex, then to cytochrome C, then to cytochrome oxidase, and then to oxygen, creating water. And then again, during that electron flow, you get the pumping of hydrogen ions in the intermembrane space, creating a hydrogen ion electrochemical gradient that is the power source for ATP synthase, and hydrogen ions flow through the synthase from the intermembrane space back into the matrix, and that powers that synthase to create ATP. And remember that you're going to produce approximately 32 ATP this way. So per glucose molecule, oxidative phosphorylation is going to produce approximately 32 ATP, and that's where a majority of the ATP is made during aerobic respiration.
So per molecule of NADH, there's just the one hydrogen, right?
Per
Like per NADH.
You're not pumping that hydrogen ion, if that's what you're thinking. You're not pumping that hydrogen ion. So, yeah. That's not the same hydrogen ion. Yeah. But you're right, NADH doesn't have just one hydrogen ion. When you reduce it, it's simply picking up one. It has more already incorporated into it. Okay. Do you guys have any questions about oxidative phosphorylation? Okay. So the next thing, and we only really need to know a few things about anaerobic respiration, and most of the things you already know. So anaerobic respiration is a different form of cellular respiration that does not require oxygen, but does require an electron transport chain. And the key difference between the electron transport chain, so that's a different electron transport chain than the one used in aerobic respiration. We're not capable of anaerobic respiration. We don't have the machinery for that. We're going to go through an example that happens in E. coli, which is a bacteria. But a key difference between anaerobic respiration and aerobic respiration is that the final electron acceptor is a molecule that's not oxygen. So what we're going to see is that at the end of this electron transport chain, the electrons are not passed to oxygen, they're passed to another molecule. And what molecule they get passed to depends on what organism you're talking about. So there's other organisms that are capable of anaerobic respiration, and the final electron acceptor can be different depending on the organism that you're talking about. So for this class, you just have to know that it's not oxygen. So in anaerobic respiration, something similar happens, that you get electrons flowing through the electron transport chain, and in this example, they're passed to nitrate. And during that flow of electrons, you get the pumping of hydrogen ions again, and that creates a hydrogen ion electrochemical gradient that again powers ATP synthase, which is then going to produce ATP during this part of anaerobic respiration. So really the key difference, the things that you need to know is that anaerobic respiration, it also includes the other parts, glycolysis, etc. It's just that the last part is different. So the electron transport chain part is different. And then the final electron acceptor is not oxygen. So this process does not require oxygen, which means that these E. coli could perform this in the absence of oxygen, which means that they can survive in the absence of oxygen, whereas we can't. What do you mean by that? Well, it's in the air. Nitrogen gas is in the air, but that's not, nitrogen gas is N2. So this is nitrate.
So like E. coli, so can it live in water?
I think so. I'm not positive about that. Yeah, but I think so. Yeah, because you grow, like when I've grown E. coli, I grow them in a broth, which is basically water plus nutrients. So yeah, they can grow in water.
What's required for anaerobic respiration, like instead of oxygen?
Well, in this case, it would be nitrates, so NO3. So rather than having oxygen being the final electron acceptor, you're going to have to use nitrate. So for this process, if you didn't have that present, which I don't know when that would be the case, like when you would have none of that molecule, but it wouldn't be able to take place without that.
So that like in water too?
Yeah.
Okay.
Yep.
Spiders use that.
I don't know. I think that all animals use aerobic respiration.
Okay. Just like that.
Yeah, insects are animals. So remember, you have animals, plants, fungi, and protists, and insects would fall in the animal kingdom. You'll learn that in 1114. So 1114. So what you do in 1114 is you talk about evolution for a while, and you also talk about ecology for a while. But then for the rest of the course, you really just go through the different types of organisms, like protists. So you start with bacteria and archaea, then you do protists, and then you do plants, and then you do animals, and you just talk about the characteristics and the different types of organisms within each of those categories. And so basically it's a lot of like describing and characterizing organisms.
So it sounds like you go your way up, like you start with like less, I don't know, complicated animals, right? You start with that.
Well, in terms of animals, yeah. So what you do is you talk about, so if you didn't know, the sponges are also animals. So the simplest animals would be sponges. Huh?
Starfish too?
Yeah. Yeah, starfish are also animals, but most people don't think of sponges as being an animal. But those are, you start with basically the simple animals, and then, yeah, you do move up to more complex. And in terms of when you talk about animals, you'll talk about different ways of acquiring oxygen, because not every animal has the same type of respiratory system that we have. So one thing, like worms, certain types of worms simply get oxygen by it diffusing into them rather than having like a circulatory system that pumps the oxygen around. Well, they do have a circulatory system, sorry. But the oxygen gets to the circulatory system by diffusion rather than by inhaling it and then passing it to the blood. So you just talk about how they do different things. Okay, so do you guys have any other questions about anaerobic respiration?
There's a process that causes like lactic acid to build up with it.
We're getting to that.
Oh, okay.
Yeah. So lactic acid gets produced during one type of fermentation, and it's the type of fermentation that occurs in your muscle cells. And you're going to produce a lot of lactic acid during strenuous exercise, because what happens during strenuous exercise is that your muscle cells don't have enough oxygen to produce the amount of ATP through aerobic respiration that you would need. And so then it also uses fermentation to produce more ATP, because you don't need oxygen for that. And then when you start running that, you're going to produce lactic acid in the muscle, and then that's going to lead to lactic acid buildup during strenuous exercise. Okay, so the last thing that we're going to talk about in this topic is fermentation, which we said is just another pathway that can produce ATP. And we can break down, so there are different types of fermentation pathways depending on what organism you're talking about. The two types that we're going to talk about, one is called alcohol fermentation, and it happens in yeast, but it also happens in other organisms. It does not happen in us, so you can't get drunk by exercising a lot. But the kind that does occur in us is lactic acid fermentation, which is the second type that we're going to talk about. No matter what type of fermentation you're talking about, you can break down fermentation into two parts. The first part being glycolysis. So glycolysis is actually a part of all three pathways that we've talked about. So it's a part of aerobic respiration, it's a part of anaerobic respiration, and it's also a part of fermentation. And the second part of fermentation is the regeneration of NAD+. Okay, so we've already discussed what happens during glycolysis. So during glycolysis, you're going to take glucose and break it down into two pyruvate molecules. You're going to produce 2 ATP and 2 NADH molecules. And we said that in that process, you're taking NAD+, you're giving it electrons, and you're reducing it to NADH. So in terms of this pathway, if you only had glycolysis occurring without the second part, eventually you would reduce all of the NAD+ to NADH. And if you did that, then glycolysis would also shut down and you couldn't produce ATP. So the second part of this pathway doesn't produce any ATP, but it regenerates the NAD+, so you can keep this part going. So during fermentation, the only part that produces ATP is glycolysis. And so for fermentation, per glucose molecule, you're only going to produce 2 ATP. That's in contrast to the 36 that you produced during aerobic respiration. So in terms of efficiency, you get more energy from aerobic respiration per glucose molecule than you do fermentation. The advantage of fermentation is that you can do that in the absence of oxygen. So if you don't have oxygen present, fermentation can still take place. Okay. So again, fermentation can be broken down into glycolysis and the regeneration of NAD+. What we're going to do is talk about two fermentation pathways. We're going to talk about alcohol fermentation. And I told you that this is the type of, this fermentation pathway occurs in yeast. And yeast are also capable of aerobic respiration. So if you grow yeast in the presence of oxygen, they're going to perform aerobic respiration because that's more efficient. But if you grow yeast in an environment that lacks oxygen, then they're going to use fermentation to produce the ATP. And yeast can actually survive on fermentation alone, whereas we can't. We need aerobic respiration. Okay, so what happens in fermentation is you're going to start with glucose. And during glycolysis, so this represents glycolysis, you're going to take that glucose and break it down into two pyruvates. Also, you already know that during glycolysis, you're going to produce 2 ATP, and you're also going to produce 2 NADHs. And again, what's happening to those NADHs, or the NAD+, is that NAD+ is being reduced to NADH. So I wrote glycolysis in here, so that's why it's not exactly connected, but this is 2 NAD+ is being reduced to 2 NADHs during glycolysis. Okay, and then what happens in this fermentation pathway, so glycolysis is the first half, that's the first half, and everything else I'm going to write on the board now is part of the second half, which is regenerating NAD+. So what's going to happen in this pathway is you're going to take the pyruvates that you make, and you're going to break each of them down, and each of them gives off a carbon dioxide molecule. And after you you break them apart into carbon dioxide and acetaldehyde. Okay, so again, you're going to break down pyruvate into carbon dioxide and acetaldehyde. So at the end of this first step, you're going to be left with 2 carbon dioxides and 2 acetaldehydes. Now, if you remember back to the lab, you guys actually measured alcohol fermentation in yeast, and you measured it by the carbon dioxide that's produced causes the airspace to grow larger in the test tube, if you remember back to that experiment. And so that's what you actually measured in that experiment, was the carbon dioxide that was being produced during this step. Also, when you talk about bread that are made with yeast, this is what causes the bread to rise with the bread that you make with yeast. Okay, then what happens in this pathway is the 2 acetaldehyde molecules that you make are going to be reduced, and the electrons that are used to reduce them come from NADH, and in that process you're going to, so acetaldehyde gets reduced, NADH gets oxidized to regenerate NAD+, and acetaldehyde is reduced to ethanol. So you end up with 2 ethanol molecules at the end of this fermentation pathway. And that's where the ethanol comes from in wine and beer. So the alcohol in wine and beer is the ethanol that's produced in this type of fermentation pathway. Okay, so in terms of alcohol fermentation, you get ATP produced during glycolysis, and then during the regeneration of NAD+, you're going to produce carbon dioxide and also ethanol as final end products of that second part. The second part, again, being regeneration of NAD+.
Carbon dioxide being exploded?
Yes. Mm-hmm. Does everyone have this written down? Because I'm going to erase it. Okay. So then we're going to talk about the second fermentation pathway, which is lactic acid fermentation. And this starts off the same way. So again, you're going to start with glucose. And during glycolysis, you're going to break that glucose down into 2 pyruvate molecules. And also you're going to produce 2 ATP. And then also you're going to reduce 2 NAD plus molecules to 2 NADH. I forgot to write the 2 in the last pathway, but don't worry about it. Oops, draw that coming out of here. Okay, then what happens in this pathway is that the pyruvate that you produce, in this case, is going to be the electron acceptor for electrons coming from NADH. So the electrons from NADH are used to reduce pyruvate to lactate, and that's the lactic acid that you produce. So at the end of this pathway, you get 2 lactates produced when you regenerate the 2 NAD plus molecules.
What makes the difference between that? Like how does, I mean, obviously like depending on what organism it is, but how does it know?
How does it, sorry, what's that?
So like you have the 2 pyruvate.
So the second half in terms of the organisms are catalyzed by different enzymes.
Okay.
So that's what's going to make them different.
Okay.
So we don't have the enzymes for alcohol fermentation. Okay, so in this pathway, you're going to produce lactate, and again, if you've heard of lactic acid buildup in terms of strenuous exercise, this is the process that's producing that lactic acid buildup during strenuous exercise. And the reason that you produce a lot of it during strenuous exercise is because during strenuous exercise, you're basically going to exceed the oxygen capacity of aerobic respiration, which means that you're going to max out. And you're also, so you're going to need more ATP than you can make through aerobic respiration. So then you're going to make additional ATP through lactic acid fermentation because it doesn't require oxygen. But in this case, if you continue to do that, that's going to lead to lactic acid buildup. Again, the advantages of fermentation is the advantage of fermentation is that you can produce ATP without oxygen. The disadvantage is that it's less efficient. So you only produce 2 ATP per glucose molecule. And so what that means is that lactic acid fermentation, in order to produce a lot of ATP, is going to have to consume a lot of glucose. And that's true for any fermentation pathway. Any fermentation pathway, if you want to produce a lot of ATP through a fermentation pathway, you're going to have to consume a lot more glucose compared to the amount of glucose you would have to consume to feed aerobic respiration. Okay. Okay, so the next slide just shows you pictures of what we just drew on the board. So this is drawing out lactic acid fermentation, and then this is drawing out alcohol, or you can also call it ethanol fermentation. Okay, so those are just the 2 pathways that we talked about. Do you guys have any questions about fermentation?
If you're exercising, how do you know your body's not so on oxygen?
I don't know that you consciously know. You don't consciously know.
So,
I don't know if there is a way. I'm not really an expert on exercise physiology, which is what you'd have to be to know that. I don't think there is because, I don't know, I think I've exercised strenuously and I don't ever be like, yep, I'm doing lactic acid fermentation now. But I don't know. So.
it hurt damage your muscles, right?
I also do not know the effects of it is so. I don't know what it does in terms of your muscles. I do know that lactic acid, because it's an acid, can lead to a pH change in your body, which is not good. So remember, you want to keep the pH of your body relatively constant. And if you get lactic acid buildup, you're making a lot of acid, which is going to decrease the pH, and that can lead to acid. It's one of the things that can lead to acidosis, which is what you would call a decrease in, obviously you know that, a decrease in pH in your body is called acidosis. And there's different causes. So one cause of acidosis could be lactic acid. There's also forms of acidosis that are caused by breathing problems. And then there's also some that are caused by kidney problems, because your kidneys also play a role in regulating the pH of your body. Do you guys have other questions? Okay, so let's do some of the lecture questions for this topic, and then we'll see what time it is and decide if we're going to move on to the next topic after this, which is cell division.
So do you think there's enough time to like catch up with all of this stuff? What happens if we don't?
We'll get to it. So if we don't, then it's, I mean, in terms of this class, it's not of a concern in terms of tests or exams, because I'm not going to test you on something I didn't present to you. In terms of actually having the knowledge, if we don't get to something, I will post. You have access to the Blackboard website until like next semester, I think till the 5th, or what, do you guys know what week it is? Anyways, you're going to have access to the Blackboard website into next semester. And what I'll do is if we don't get to a topic, I'll post a lecture for that topic so that if you want the knowledge, you can go on and listen to it. And I'd be more than happy to answer any questions that you have so if you watch a topic, we're obviously not going to have lecture, but you can always email me and I'll answer any questions. So that way you at least will have that knowledge because it's something you are supposed to learn during this class. But in terms of being asked on quizzes and exams, it's strictly going to be what we cover. Okay, so let's do this first one. So basically I want you to explain how the following interact to produce ATP. So explain the relationship between a hydrogen ion electrochemical gradient, the electron transport chain, and ATP synthase.
The electron transport chain fuels the hydrogen ion electron in electrochemical gradient.
And by fuel, what do you mean?
It produces.
Right, produce. So the electron transport chain produces the electrochemical gradient.
Which in turn fuels the ATP synthase by adding hydrogen ions.
Right. So again, the connection is the electron transport chain creates the electrochemical gradient, and then the connection to ATP synthase is that the electrochemical gradient is the power source for ATP synthase, and you stated it correctly that the hydrogen ions are going to flow through the synthase to power it. Okay, so remember we've divided aerobic respiration into glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation. Which stage produces the most ATP? Right, oxidative phosphorylation. Which is the stage that actually requires oxygen? Oxidative phosphorylation, right. That's where the oxa part comes in oxidative phosphorylation. Okay. So number 11, this isn't something I explicitly stated, so you should be able to guess what the answer is though. In aerobic respiration, carbon dioxide is formed from the oxidation of the following. So remember you're reducing NAD plus and FADA molecules during this process. Where do those electrons ultimately come from that are being used to reduce those molecules? No? That's for, so water is the electron donor in photosynthesis. What's that?
Glucose.
Right, glucose. So ultimately the electrons are coming from glucose during aerobic respiration, and those electrons that are stripped from glucose are going to be given to either NAD plus or FAD, depending on what part of aerobic respiration.
Because everything's in glucose, right?
Right, so yeah. So basically if I asked you, ultimately, if nothing else happened in the cell, so one thing I didn't tell you is that these pathways connect to other pathways within the cell, so you can use some of the molecules for other things. But let's just say that you were only running this pathway. Glucose ultimately ends up as what? The carbons in glucose ultimately end up as what? Right, carbon dioxide. And then the electrons all end up in oxygen. Okay, So 13, use the following word bank to fill in the blanks below. So part A, during oxidative phosphorylation, blank and FADH2 are oxidized to power ATP production.
NADH.
Right, NADH. B, the electron transport chain is located in the inner membrane of the mitochondrion. C, the final electron acceptor of the electron transport chain is oxygen. NADH and FADH2 donate what at different points in the electron transport chain? Right, electrons. And then ATP synthase uses a blank to produce ATP.
An electrochemical gradient.
Right, the hydrogen ion electrochemical gradient. So the full thing, a hydrogen ion electrochemical gradient. And again, the way that it uses it is that the hydrogen ion electrochemical gradient, again, is the power source for that synthase. Okay. This one, again, it's not something I explicitly told you, but you should be able to, knowing the pathways, be able to figure it out. The oxidation of NADH results in the formation of more ATP molecules compared to the oxidation of FADH2 because why? No? No? It's A. So if you remember the electron flow, the electrons coming from NADH pass through 3 hydrogen ion pumps, which means you're going to get 3 different pumps pumping hydrogen ions, whereas the electrons from FADH2 only pass through 2 of them. So you get the pumping of more hydrogen ions in terms of the electrons coming from NADH than you do compared to FADH2. Okay, 15, before entering the Krebs cycle, pyruvate is converted into what? Right, D, acetyl coenzyme A. 20, which of the following is true of the citric acid cycle? Right, that it produces ATP, NADH, and FADH. Let's just talk about the other ones really quickly. So it doesn't yield any NAD+, it produces NADH. So it yields NADH. And then there is no part of aerobic respiration that produces oxygen. Okay, so no part of aerobic respiration produces oxygen. Only part of it, it's only consumed, and it's only specifically consumed in oxidative phosphorylation. And then it doesn't occur in the inner membrane of the mitochondria, it occurs in the mitochondrial matrix. Okay, so 21, which of the following function location relationships is incorrect? Right. Where is acetyl CoA formed? Right, because acetyl coenzyme A is produced by pyruvate oxidation, which we said happens in the mitochondrial matrix. So acetyl coenzyme A formation should be matched up with the mitochondrial matrix. Okay. 25, this is basically repeating a question we've already asked, but it's an important one. High energy electrons from molecules of NADH and FADH2 are transferred to an electron transport chain. What is the final electron acceptor of this electron transport chain? Right, B, oxygen. And again, that's where you're consuming oxygen on this process. So 29 and 30 get at the flow of electrons. Just the one, oh no, that's fine. So for 29, which one correctly states the pathway that electrons take from NADH to oxygen? Right. So it's going to start, they're going to get past the NADH dehydrogenase complex, then to ubiquinone, then to cytochrome BC1, then to cytochrome C, then to cytochrome oxidase, and then to oxygen. What about basically the same question, but now we're talking about the electrons coming from FADH2? It's not C, B, right, B. So you're going to start, and the electrons are first past the succinate reductase, then to ubiquinone, and then the pathway is the same from ubiquinone, cytochrome BC1, cytochrome C, cytochrome oxidase, and then to oxygen. Okay, 32, sports physiologists at an Olympic Training Center wanted to monitor athletes to determine at what point their muscles were functioning anaerobically. They could do this by checking for the buildup of which molecule? Right, B, lactate, because that's going to be produced by lactic acid fermentation, and that's what you're going to be producing when your muscles are functioning anaerobically, which means without oxygen. 33, which of the following is true of ethanol fermentation? Not F.
C.
Not C. It's A. Yeah, A. So pyruvate is converted to acetaldehyde. So remember that this happens during the regeneration of NAD+. So the first thing you do in alcohol fermentation, once you produce pyruvate, is you break it down into acetaldehyde and carbon dioxide, and then the acetaldehyde is reduced to ethanol. This one, C is not true because you don't get any net production of NADH, because all the NADH that you produce in glycolysis gets regenerated into NAD+, so you don't get any net production of NADH. And then E, carbon dioxide and ATP are required, so that's not true. Well, is ATP required?
Yes.
For what part? Right, glycolysis. So remember, the energy investment phase of glycolysis does require ATP, but carbon dioxide is not required. It's a product of the pathway. So it's not a requirement for the pathway. And then D, oxygen is required to break down glucose. So that's false because we discussed the fact that fermentation does not require oxygen. That's because you're only using glycolysis to produce ATP, and glycolysis on its own doesn't require any oxygen. And then NADH is used to reduce pyruvate to lactate. That doesn't happen during this fermentation pathway. That happens during lactic acid fermentation. Okay, do you guys see other questions that you guys want to go through from this topic? 16? Right, okay, so a single turn, so let me ask this, how many times does the Krebs cycle turn per glucose molecule? Twice. So this is asking for only one turn of the Krebs cycle, what does it produce? Right, C, 1 ATP, 3 NADHs, and 1 FAH2s. And remember that when I wrote the products on the board, I was writing the products per glucose molecule. That's why I wrote 2 ATP, 6 NADHs, and 2 FADH2s, because that cycle turns twice per glucose molecule. Yep.
Does the CO2 not count carbon dioxide? Because I just feel it's this because you have 4.
Oh yeah, that's just not mentioning that as a product. So that's just talking about the energy intermediates that are produced. They just didn't mention. These questions are taken from a question bank, so yeah. Other questions that you guys want to go through from this topic?
What was 7 again? Remember you going over it? I think I wrote it down.
Oh, I didn't. I mentioned it when I was lecturing, yeah. So one of the components of the electron transport chain in the mitochondria is inhibited by cyanide. Explain why this would be detrimental to the organism. So remember that if you inhibit the electron transport chain, which is what would happen with cyanide, You won't make the hydrogen ion electrochemical gradient. If you don't make the hydrogen ion electrochemical gradient, there's no power source for ATP synthase, and then you can't make ATP that way. And remember, you're supposed to be making about 32 ATP with that synthase, and so you're basically, if you take cyanide, you're going to be preventing your cells from making a majority of the ATP that is made during aerobic respiration. And so basically, if you inhibit it, you'd only be able to produce ATP through the Krebs cycle and the glycolysis, and that's not enough ATP for us.
Do you know the rate at which the average human uses ATP?
They probably do know that, but I don't know. It's a lot.
I would imagine.
Yeah.
So, like, this is happening like really quick.
Oh yeah.
All the time.
So one question that you guys didn't ask is, so ATP is not very stable, and so that's why you can't store ATP. That's why you can't store energy as ATP, because ATP is an unstable molecule. So that's why you have to constantly produce it, rather than producing it and storing it. You store it as, like in us, as glycogen, which you break down to glucose, and then you break the glucose down to produce ATP. So yeah, it's something that's constantly happening.
So when we're talking about the electron transport chain, we're not talking about each electron being individually passed. It's like a flow, sort of like a
Yeah,
current.
it's yes,
It's just happening.
yeah, yeah, and that's constantly happening. Yeah, yep.
Or just happen at? I mean, it's not every cell. It's
Every cell of your body. Every cell of your body. Well, okay. I don't know about red blood cells. Not sure, because red blood cells also don't have a nucleus. Because when you produce them, they're produced from a cell that has a nucleus, but red blood cells themselves don't have a nucleus, and I don't know whether they have mitochondria or not. But every other cell in your body, I know, definitely has mitochondria, because every cell in your body needs energy. Every cell in your body needs to make ATP. So now, depending on the cell type, you can have different numbers of mitochondria because there are different energy requirements. And in terms of turkey, that's the difference between light meat and dark meat. So what makes dark meat dark is the fact that there's a high amount of mitochondria in those cells.
So are there like any, or is that,
Okay.
So that, I mean, that's because the energy requirements for 1 round are required in the leg meat.
What's that?
So like the energy requirements
Well,
for 1 round from the leg muscles, so the leg muscles need.
Yeah. Well, so within muscles, it's a little bit confusing because there are actually, so you call a muscle cell a muscle fiber, and there are different types of muscle fibers, and it depends on what do they mainly use to make ATP. So one type of muscle fiber uses mainly glycolysis to produce ATP, and then other ones will use mainly oxidative phosphorylation, and those are the ones that are going to have a lot of mitochondria in them. The ones that produce it mainly by glycolysis are going to have an abundance of the enzymes that are involved in glycolysis in the cell. Yep.
How can a little bit of like cyanide kill you, like all your cells? Like I feel like it wouldn't be enough material for all the cells to die.
Oh, I don't know. I don't know what amount is needed. I don't know what dosage. Yeah, I don't know the dosage for the amount of cyanide you'd have to take to kill you. I have no idea. I'm not an expert on cyanide poisoning. What's that? Yeah. I don't know. I don't know if you can inhale it or not. I don't know. Yeah. Well, I mean, I think you definitely would have to. Well, I mean, yeah, you could. If you inhaled it, it'd probably get to the rest of your body through your bloodstream. So yeah, but I don't know what dosage is required in order to cause the death part.
But I mean, it stops, like you need ATP that bad. I mean, it stops it. Obviously, your body just gets out. I mean, that's how fast you're burning.
Yeah. Because you can't store any ATP, so you have to constantly be making it.
Are there other things that stop ATP production that like we
That you know of?
go through like daily or something?
Not off the top of my head, I don't know. Do you guys have other questions? Okay, so we'll stop there for tonight. And then remember that on Wednesday, you do have the pre-lab due, but you also have that second experimental design.